for the invitation. It's a pleasure to be there, I will say, whatever, to be to be able to tell you about our work. And it's a pity that it's not in beautiful Bologna. Let's hope for better times. So let's see. So for today, I thought in telling you about the recent work we have been doing in studying the relation between diseases. And this is the work of two PhD students, John Sanchez Valle and Beatriz Urda. So essentially what I'm going to tell you is what they are doing for their PhD. Obviously, there are the contribution of other people in the lab, David Alba Lepore, and others, but they are the main authors of the work I'm going to tell you about. So disease comorbidities, just to start with some definitions, is the co-occurrence of two or more diseases in the same patient. Obviously, we see this as an interesting system, interesting biological process, why people with one disease tend to have more chances of having a second disease. What are the molecular basis for that? What are the possible genetic basis for that? How this happens at the molecular level? At the same time, this has obvious social and medical implications. We see with the age, you get more chances of having more diseases. Elderly people tend to accumulate a number of diseases. And we'll say the medical systems in general are not well prepared to deal with comorbidities. They are, in a way, medicine is organized, is more by specialties addressing one disease. And when there are combination of diseases, there are less specialists treating this combination of diseases. It's a little bit like in science, if you are very well placed in a given area, normally it's easy for you to pass the evaluations because you know the people, your colleagues and all that, and you know the area. If you are working in some kind of interdisciplinary research, then you have problems because nobody really understands and you belong to different areas. So with diseases, it's a little bit the same thing. The medical systems are not very well prepared for treating diseases, and this becomes a real social and economical problem. So a few years ago, we decided to enter in this area and try to understand the relation with diseases at two different levels. At the, we say, more medical level, how we can follow the occurrence of diseases in the medical records of real people, and at the same time trying to understand what are the molecular bases, or at least some of the molecular explanations of comorbidities. Obviously, we are not the first ones working in this area. There are plenty of papers dealing with relation with diseases. The two main groups, I would say, that have been producing very interesting work in this area. One of them is Laszlo Barabasi and colleagues. Laszlo Barabasi is a very famous physicist working in networks, and one of the networks that he has been studying is the network of interaction with from diseases. This, in his case, a number of papers are based on insurance records from American system, some of the American systems, and essentially what he's drawing is these beautiful networks. The colors correspond to different type of diseases. They're classified. The lines between them means that these two diseases are related. And then there are this clustering and classification of diseases and classification of the diseases and the relation with the what they are related possible phenotypes. The other group that has done seminal contributions to this area is the group of Søren Brunak, and in Denmark, a good friend and a frequent visitor of the Bologna School in the old times. He's using the Danish medical records. In Denmark, to the contrary of other countries, it's easy to access, for scientists, easy to access the medical record because they are considered a social asset and something that has to be exploited for the social good, what makes things easier. So he has been deriving relation between diseases. So each one of these small balls will be one disease in a number of patients. The lines or the arrows connecting them is how frequently these two diseases are connected. And what he has done, and is particularly interesting, is to derive this in terms of trajectories. So from one disease to the other, what is the probabilities? And there are different paths in this disease. You can go from one disease to another or to another ones, and depending on the path you choose, the consequences could be different. And all this is based on statistics extracted from the observations in the medical record, from the diseases code in the medical records. So we went to this, trying to understand these relations, trying to get our own data on these relations and understand these type of relations. And I would say the particular thing that we have been doing, show you in a few slides, is trying not to see the problem at this very general level of all the connections between diseases, but trying to see this problem more taking into account age and gender. And you will see in a minute that, and I guess makes a lot of sense, diseases are not the same for males and females. Diseases are not the same for young people and for less young people. So the data we have is from Catalonia. This is I would say, well-organized medical system, around 5.5 million people that have a unified system for primary care. So always that you go to the doctor, there is a registry. So they have millions of registries of each time a person goes to the health system. This accounts for a very large collection of medical records. So in these cases, the diseases are coded in ICD-10. This is one of the ontologies or one of the systems for coding diseases at different levels of granularity. So we are using this classification at a given cutoff, the one that is given here for maintain diseases at this level of classification. We calculate the relative risk. This is a technical note, so this is a way, I would say, is well established in the field of calculating, given a type of disease and a number of observations of people that have or not this disease, how, if they have a second disease, what is the relative risk from having one disease to have a second disease. And this is calculated in terms of the proportions of patients that go from one disease to the other, respect to the total number of patients that go from one disease to any other disease. There is an important difference here between chronic and acute diseases. Acute diseases, as a definition, we use something that happens only in 1 year and is not repeated in the following years. Chronic diseases are the ones that happen in a year, and then they are extended for a very long period in a way that we don't count a co-disease with a chronic disease many times every year. We just count this is something that is happening along all the time. Just to make this clear, because this has been an issue that has been confused in some previous publications and then becomes a little bit of a mess if someone with a disease for many different years, every year or every time, you count this as a new observation. So we are talking with a network that has around this number of edges and nodes. So we are talking about the thousands of nodes around for in total. There are different ways of representing that, just to give you an idea. So this is 2 different networks for males and females. I think that's the point I want to make here. For one disease, type 2 diabetes, and how this is affecting, correlated, statistically speaking, with other diseases. And first of all, the networks are different. Males have many more diseases correlated with diabetes. Females have less. In general, you will see females are more healthy than men overall. And the relations are different. So there is a clear relation in men with Parkinson that is not so clear. There is a clear relation with kidney cancer that is not in men. So once that you start observing any disease, you will see that statistically speaking, males and females behave in a different way. Obviously, you don't want to say this only as a very general level, because there is a second implicit bias, that is the age. The distribution of ages in the males and females are very different. Females live longer, and usually the curve is flat because of that, because it's extended towards older ages. So you have to take into account males and females, but you have to take into account also the age distribution. These are some plots indicating that. I don't want to go into a lot of this details here. These are males and females, and this is the accumulated number of diseases using, again, as an index, type 2 diabetes. And again, there are some obvious differences. First of all, the number of comorbidities is larger in males. The distribution of the colors is respect to the different type of diseases is also very different. So the relation between age and gender is important as a bias that has to be taken into account. These are the same thing, males and females by age type, having the first disease or a second disease. And this is, again, important because the distribution of the first incidence of the disease or the probability of having this disease as a secondary disease is also very different. So in summary, without going to specific details, of which there are many, and they are interesting for the medical consequences, is I would say that it's interesting that while the analysis of Soren and Barabasi are very interesting as a general level, if you really want to say something interesting at the medical level, you have to go into at least these 2 co-variables, age and gender, and do this analysis specifically for age group and sex, because otherwise you are mixing very different things, I would say. This problem of the biases for comorbidities is important, but it's important for many other topics, a part of the social ones, scientific ones. So in general, all the data set that we are getting don't have a very clear distinction between males and females, what makes that the conclusions are, in a sense, a mixture of different things. And the example of comorbidities, I think, is a good example in this case, because what we see is that you cannot really mix the 2 sexes for this analysis. And this is true for many other things that are done at different levels of analysis of data set. Genomics will be a good case in which if the data set are not properly labeled, it's difficult to distinguish with influences coming from gender or for other issues. So I leave this chapter here. So we can analyze the population, the Catalan population. We can reproduce many of the things that has been observed by Barabasi, by Soren Brunak and other groups in terms of diseases, but then we want to make not a general observation, make these observations very specific by gender and by sex. And I'm not entering the examples, but then the full thing is to find the specific cases of comorbidity, the specific trajectories that are particularly different between males and females. Now, the second question, very much related, is, okay, this is fine. This is observational data. We see that a person with one disease have more or less chance of having another disease with time. Why this happen? Why having one disease may increase the chances of having another one and not others? And why diabetes increase chances of some cancers and not others? Are there some, is this related with some basic molecular mechanisms that may be behind these susceptibilities from related with different diseases? There are different ways of doing that. The way we have been doing this is to analyze expression profiles. So essentially, it's pretty simple. What we do is to take different diseases, go to a large scale data set. These are RNA-seq studies, gene expression by sequencing of different diseases. We find the different studies, we create a pipeline, we derive what are the genes that are more or less expressed, highly expressed, or the expression is reduced. Trying to do this properly, taking into account that different studies have different biases, accounting for that, and trying to do the bioinformatics properly. And then we analyze the relation between the different diseases, comparing the expression profiles of the genes between different diseases on different individuals. So that's not going to enter into the technical details, filtering, normalizing, doing differential expression, and then doing comparing the differentially expressed genes, and doing functional enrichment and gene set and pathways. So this will be the type of results in this area. We have on the left, the colors correspond to the different diseases classified in terms of ICD codes in this ontology. And the x-axis, you have different type of diseases. And this is showing the overall correlations. Blue means these two types of diseases are strongly correlated. Red means they are anti-correlated. And this is something that is interesting. By comparing the expression profiles, we not only find, and this done, at the level of pathways. So they are comparing ICD codes and reactome pathways. The positive and the negative is interesting, because this means that there will be pathways that are overexpressed in a given disease. So they tend to be, genes in these pathways tend to be overexpressed, and there are pathways that tend to be repressed in this for a given collection of diseases. The blue represent this overexpression, the red represent this downregulation. And then you can spend time in analyzing these details. I showed you just a couple of examples of these. These are for two different type of functions. Functions related with neural systems, or pathways in reactome related with neural systems, or pathways related with the structural matrix organization. So two, in principle, different biochemical or biological systems. And these are a number of diseases related with neurological or neuromotor diseases. Schizophrenia, autism, dystrophies, Huntington disease, and Parkinson's disease. The interesting observation here is that even if some of these diseases are classified at the level of diseases in similar ways, the behavior at the level of pathways is very different. In some of them, the ECM is activated, in some of the others is not, in some of them there is a downregulation of the neural systems, in the other ones is not that obvious. So the molecular characteristics of the diseases brings a different type of information to the one that you can do at the level of the diseases. So in a sense, we have a more fine-grained way of defining diseases at the molecular level. This will be another example. These are different types of colitis, ulcerative colitis, ulcer Crohn's disease. And again, it's the same thing. Even if the diseases are similar from the medical point of view and classifying the same sector of ICD codes, their implications at the biochemical level, at the level of the biochemical pathways, is different, meaning that the molecular mechanism behind it, analyzed these ways at the level of gene expression, may be different. Using this information, obviously, we can compute a disease similarity network. In this case, what we are doing is comparing the profiles of the cases classified in the different diseases to see how they relate one with the other. So again, in this case, the blue dots will mean a relation between diseases, and the red ones will mean an anti-correlation. And that's because an interesting topic from the medical point of view and from the biological point of view, indicating that it's not only that by having one disease, you may increase the chance of having another disease, it also can work the other way around. Having one disease may decrease the chances of having another disease. This protective, you can call it the protective effect of the diseases, is well documented in the literature. There are observational examples of following a group of people with one disease that have less chances of having one other disease. The interesting thing, obviously, here is that we can see that at the level of the expression of the expression profiles. Our inference in this case will be that having expression profiles that are behaving in the opposite way, in one disease they are increasing, in the other one is decreasing for a given biochemical pathway, may be correlated with these observations of the diseases that are anti-correlated. Having one disease decrease the chance of having another disease, opposite to the cases in which having one disease increase the chances of another disease that will be seen as a positive correlation between the corresponding pathways. Well, these are some examples, the inverse comorbidities, and bowel disease and lymphocytic colitis that have different correlations between different diseases, even if the secondary disease associated to that will look like similar, they may be specific correlations. Another way of saying that is that for a number of cases, we can go to one disease and make a more specific classification of what are the potential correlation with other diseases by analyzing the expression profiles. So these are the observations at the molecular level comparing expression profiles. So what we are saying here is about the expression profiles. The real question, I think this is a fundamental question, at least in my opinion, is, does it make sense? All what we see at the molecular level, apart of finding examples, are we really able to say something about the diseases, analyzing the expression profiles? Or in other words, can we use the molecular profiles to say something about the relation between diseases? Is there a correlation between our observations and at the molecular level and the medical observations? Are the correlation between expression patterns telling us anything significant about human diseases? That's, I think, otherwise, we may be only working with expression profiles that may have something to do with some biochemical functions, but they may not have anything to do with the correlation between the diseases. And the truth, it is correlated, and I think that's the important observation. So what we are doing here is to go to this network of correlations, positive and negative correlation between expression profiles, and at the same time checking for each one of them if they are, or if they have been seen or not at the level of diseases. And to do that, we are taking this Hidalgo et al. Is the Barabasi network. So it's based on real observational data. And for almost 50% of the interactions we observe, they are also seen in the medical records used by Barabasi. If we take only the diseases that have enough observations, that don't have enough differential expressed genes, these correlations become even higher, 60%. So we'll take this number as a more significant one. These two, I mean, for me, this is I'm still surprised by these numbers. So that's something that is essentially very simple. Comparing expression profiles can tell us about the correlation between diseases. Is first of all, is surprising, and second is has I would say important biological implications. Just analyzing expression profiles, we can have an idea of what are the relations at the disease level. That is a completely higher up level of the phenotype. So that's I would say that's the observation, and that is the implication. As I said, for me is important. Expression profiles of patients contain enough information to reproduce many of the comorbidities that are known at the medical level. So it's a tool that we can use to explore comorbidities. We can do more than that. This is just the general analysis of the diseases at the level of general disease, but obviously we have individuals. All these diseases, we have observations at about specific individuals. So we can now go and go from the general disease to group of cases of a given disease that have similar characteristics. What we call meta patients are an aggregation of the patients of a given disease that behave in a very similar way at the level of the expression profiles. So that's, in this case, we'll be processing the data in a way that we can obtain from all the patients of each one of the diseases and all the correlations, these meta patients, by doing clustering at the level of the patients. So that will be an example, just to make clear what I mean here. This is breast cancer. As you know, there are well-known subtypes of breast cancer. And when we analyze the subgroups using the information of the comorbidities, we reproduce the groups that are already known at the level of the disease. So we have enough information dealing with the expression patterns and the comorbidities and the correlations with the other expression patterns to reproduce the classes that are already known. So now we can do these meta patients that will be an aggregation of the different type of subtype of cancer and study not the correlation between breast cancer and other diseases, but the correlation of the patients of the subtypes with the other diseases. And then obviously once that you do that, you observe things that are different than the ones you observe when you put all the data together. So in this case, the one that is highlighted is the relation with autism that is positive in 2 cases, but is not present in the other subtype of the breast cancer. So again, a little bit like in the case of the gender, once that you analyze the data in a more precise way, making smaller classes, you can observe things that will not be visible when you do the observations at the general level. Well, this is obviously, there are plenty of examples that can be followed and analyzed. We have a tool that allows to do that, and this is, without entering in a lot of details, is a tool that allows to analyze the disease network at the level of general diseases or at this level of the subgroups of one disease in relation with other diseases. And that's if you take all the networks. You only want to see positive, or you want to see all the relations, and you can choose the level of correlation that you want to see in the network. You want to see only things that are very, very strong correlations, or things that are more weak correlations, and then you can analyze the specific cases, analyzing the relations. What are the relations between these diseases? In which data is this based? And how can you move the networks at different levels? So that's so I leave this part here. So to something that I still find surprising. We can use gene expression profiles to compare diseases, and this comparison between gene expression profiles tell us something about the real correlation between diseases. We observe at least a high number of the known diseases that are correlated. After one disease, you get the other one. We can reproduce this at the molecular level, and then obviously one that we can reproduce this at the molecular level, we can go and see what are the pathways, functions that are related with these comorbidities, and we can do this for positive and negative comorbidities, for relations of direct and indirect correlations, what opens a very interesting area of research of why some diseases are preventing other diseases, and we can do this at the general level of the diseases or at the level of these meta patients in a more particular in-depth view of the relation between diseases. There is another aspect that I think is interesting and I would say a little bit mysterious, or a little bit intriguing. One thing is to use the gene expression profiles, in which we are essentially saying how these two diseases or two individuals correlate these expression profiles, meaning if they have the same or correlated overexpressed genes and correlated downregulated genes. One thing different that we have been analyzing is gene expression variability. Gene expression and variability is a different way of looking at expression, in which we are not looking in a given individual to how many genes are overexpressed or downregulated. We are looking at the variability, meaning that between two individuals, for example, they tend to have different genes up and downregulated. A very variable gene will be a gene that in a given population, in some individuals is highly expressed, in others is downregulated. That will be the typical, prototypical variable gene at the level of gene expression, will be a gene that has this type of very diverse type of level of expression in a given population. So we have been studying this previously. It's an interesting area of research. Why this happens? Why there are genes that are, have this sort of apparently random behavior? You can think that because they are only random, or you can think that they are there because they are selected to be random. And I guess bacteria are a better example for that, because we know the typical example will be, E. coli culture. You put some antibiotics, and then some genes make that some of the individuals of the bacteria will survive to the antibiotic, because they are not all of them expressing the same genes at the same level. They have some variability, and this variability in the expression of some of the genes probably is what prepares them to have some genes that confer resistance to some of the individuals in the population. So that will be an example of something that is in a sense is embedded in the system to be able to be prepared to face change in the environment. So there is an interesting question that I'm not going to enter today in what is the selective pressure that is put in the genome or the epigenome to maintain some genes as variable. We played previously with these ideas, finding that these are chronic lymphocytic leukemia. There is a more aggressive type of the disease, and this more aggressive type of the disease is characterized by having more of these variable genes. Aggressivity means being able to escape from the control of the immune system. So it makes sense that the more variable genes, confer more capacity to escape to the variable conditions imposed by the immune system. That's the theory. So part of the problem is how to calculate differential variability, and this requires some care, because for a gene that is expressed at a very low level, it's very easy to be variable, because just expressing 2 times more will be very variable. For a gene that is expressed at a very high levels, to be very variable is difficult, because you have to express a lot of an enormous quantity to duplicate the level of expression. So you have to balance the expression to calculate variability. You have to be able to account for the level of expression, and this is why the formulas are including a coefficient of variation in a way that you can calculate this variation with respect to the expected distribution of the expression of the gene. And there is a methodology to do that, and there is a sort of agreed formula on how to calculate this variability of expression based on the expected distributions as a coefficient of variation. I skip this one. So now the point is back to comorbidities. So now we have a way of calculating not if the genes are in between 2 patients, they are highly or not highly expressed. What we are asking here is they have similar variable genes. So we are not asking for 2 patients they have this gene overexpressed. We are asking to have this gene belonging to a class of variable genes. So now we are comparing the diseases, and again mapping these 2 pathways, the same thing that we were doing before, but at the level of variability of the expression, a completely different view of the problem. Again, I don't, I'm running late, and I don't want to enter in a lot of examples. The example here are, for example, the one I highlighted, are for Crohn's disease and ulcerative colitis, representing 2 different ways of altering or not the related immunosystem pathways. So we can find specific examples in which this correlation between diseases at the functional level in terms of coefficient of variation has some implications for the type of comorbidities and can be related with what we know about these diseases. The interesting aspect of that, and this is still work in progress, is that in the same way that using the level of expression, we can find a correlation between the level of expression and the real diseases, we also find a significant correlation between the variability in these variable genes and the real diseases. So, and this is this 31%. So that means that for some diseases, we can explain at least part of the reason why they are correlated, because they have similar expression profile, they are expressing the same genes. But for some other diseases, the correlation between them may be related with the fact that they are influenced by genes that has very variable level of expression in the population. And that's, I say it's a little bit intriguing and a little bit mysterious how to translate this, and it's a little bit difficult to think on this in molecular terms, but this is the observation. So, so if the first conclusion was that diseases can be traced at the molecular level using expression profiles, the second conclusion, that is a little bit newer and more difficult to understand, is that other diseases can be traced at the molecular level analyzing variable genes, these genes that are expressed at different levels in different members of a population, different cases of a given disease. How do you interpret this at the molecular level? Why these genes and pathways that correspond to these variable genes are variable, and why they are influencing the diseases is an interesting subject of discussion, for which I don't think we have all the answers at this point. but we continue thinking on that. So to finish, I will go quickly because I'm running a little bit late. On another aspect of this relation between diseases, seen from a different angle. So one of the reasons that we are arguing that are behind the relation between the diseases, behind the comorbidities, is molecular basis, that they have similar expression profiles. Therefore, there may be a genetic reason why people tend to have this gene more expressed, and this create chances of having one disease, but also chances of later getting some other diseases. That will be something that is part of the genetics, whatever these are, because of a genetic constitutive factors, mutations, or because of environmental factors, alterations of the level of gene expression that goes to the epigenetic level, and this is why they have these particular genes up or down regulated. The other reason that may be in the middle of all that is that the reasons of some of this correlation between diseases may be related with the drugs that some of these people are taking in some of the diseases. So the hypothesis here will be that people with one disease are taking for a long time a given drug for the disease, and this drug in the long run may have a positive or a negative effect in creating a second disease. Positive meaning in the sense of creating a higher chance of getting a second disease, negative meaning that maybe because they are taking this drug, they have less chances of having another disease. This is an interesting subject. We are not done with this at all. So we are not. I'm not going to show you data on that because we have not finished with this. But once that we started thinking in drugs, we start trying to understand better how the administration of drugs is working in the system and how it's reflected in the medical records. And the problem that I'm going to show you a few slides about is an interesting problem, and a problem that becomes more interesting as you get older, but I will show you data that is also interesting for young people. Is the problem of co-dispensation. So, you go to the doctor and you got a drug for one thing, and then you have a second disease or second thing. You go to another doctor, you get another drugs. Are these drugs compatible? Is anyone taking care of making sure that these drugs are compatible? If you go to I go to my parents, my mother, and she's taking a number. She's 94, and she's taking a number of drugs. Is anyone taking care that these drugs are compatible between them, and how this is working? So we have done this in collaboration with a group of Luis Rocha, with Rion and Luis. They have data from Blumenau, that is a region in Brazil from which they have the medical records. And Luis and Rion were working, or they are still working in Portugal, but also in Indianapolis. And they have the medical records from a number of hospitals, health provider in Indianapolis. So with plus our medical records from Catalonia, so we have these 7.5 million people from Catalonia, data for a population of 300,000 people from Blumenau, and almost 800,000 people from Indianapolis. So we can, in a sense, we can be sure that it's interesting compare how these very different systems, two public systems, one private system, one rich country, one not so rich country, compare in terms of the administration of drugs. For the Catalan system, we're back to this the population data that I showed you before, the Catalan population. We are talking here about a set of 1,300 drugs that are used in the Catalan system. what we are looking here is a co-dispensation without entering a lot of technical details. We are looking for things that are administered in the same period. These are the drugs that are taken from the counter. So what we can see is the people are taking these drugs in the same time window of a month, and that's the way the system works. And again, as in the previous case, it's very important to take into account gender and age, because drugs are very administered in a very different way for males and females, and depending on the age. So these are, just to give you an idea, these are the combination of drugs that are given to the Catalonia and Blumenau populations in numbers big enough to be significant in the different combinations, and with the statistics of these administrations. And then you want to know, we calculate a number, a parameter to reflect how many people are taking the drugs in combination, to say how significant is the combination respect to how significant is the administration of the drugs independently. The interesting thing, obviously, is that even if there are two very different populations with two very different medical systems, there is a significant overlap, and these are the pairs of drugs that are administered in Catalonia that are also administered in a significant way together in Brazil. That's good, because meaning that even if the systems are very different, they are tending to do very similar things. So there is a significant coherence between the systems, and then there are interesting differences. Okay, more data about the significance and differences. So we can, this is just to give you an example. I'm not going to give a lot of examples because let's move to a different area. These are two drugs, one an anticonvulsant and an anticoagulant, and they are administered together. The lines means that there is a higher risk in males or in one population, and also they are administered in the Blumenau population. These two drugs should not be, in principle, administered together, because they can create bleeding, nausea, vomiting, allergy reactions. So they are anti-recommended to be administered in combination. They are still. I don't want to go much into that. They are still done. It's still done. They are still administered for whatever, for good or not so good medical reasons. But that's the situation. So and at the same time, this is happening in the two populations. So that's the type of scenario that you can imagine that you can now go and do and analyze for different diseases. Again, as in the case of the diseases, the comorbidities between the diseases, the differences between the population and if you analyze this by age, are interesting. The one that is highlighted here is a difference that is reproduced between the three populations, between males and females at the early age, in this age of this range of ages between the 10th and the 30s. So they are a little bit like in the other case. It's important to do this analysis separating by ages. To be sure of what is the significance of that, obviously you need to develop a random model that you can compare. So essentially, this is shuffling in the network of interactions, the drugs, to be able to see if these differences that we see in a given age are significant respect to the distribution of diseases. That will be the case for the ones I pointed before, between the early ages, between the 10s and the 30s. Examples, these are for, again, comparing Catalonia and Indianapolis. there will be, these are the the drug-drug interactions on the y-axis, the edges on the x-axis, and this will be the administration of two drugs, an anticonceptive and antibiotic. And this is the one that is highlighted on the left panel of Catalonia. This is significant in the Catalan population, and it's not a good, it's not a recommended combination, because this include a risk for pregnancy risk. Why this is done, unclear. One possible reason is that in Spain there is an abuse of antibiotics for all kinds of things. So that's maybe a bad reason for this to happen. Well, there are many. There are many more examples of different drugs that are administered together at a given age range for males or females in a different way, and there is an interpretation of why this happened and what are the possible risks of this medication of the two drugs at the same time. I think it's important. I'm not going to enter into the details, and I left this in the presentation. It's not that it is always. No, let me say this in a different way. This administration of drugs prescribed together that shouldn't be in principle prescribed together is done in many cases because there is no alternative. You have to treat two different diseases, and these are the only available drugs to do that. Therefore, it's not done without the doctors knowing the risk. Maybe they know the risk, and they estimate that the risk of medication is less than the risk of no medication, and then they run into these double medications. Well, these are more examples. Not going to enter into that. So this is again a movie, just to make clear this point of how the number of indications of co-administration of drugs is increasing with age. So on the left is a panel of all the drug-drug interactions for Catalonia, and on the right is how this is evolving with age. These are 10 years, and you go to the 20s, and there is an increase, 30s, there is an increase, and then you see how some of the drugs combinations are increasing more. These are the ones with the bigger circles, and then by 80, 88, or 90, there is an interesting decrease in the number of drug combinations after the 90s. Probably at this point, the doctors start thinking that maybe if the people have survived until this age, maybe they can reduce the medication, because it's not going to, they don't have to cure diseases that are not going to happen in this at this point. But the increase until the 80s is pretty spectacular, I would say. So I was saying that sometimes this another way of thinking on that is that they may be alternative treatments that with the time are appearing, meaning that in principle, two drugs are administered together because there is the only solution, and you have to treat the two diseases, because it will be worse not to treat them than to treat them, even if there is a risk in the co-administration has a risk. But then with the time, there are other drugs appearing that maybe belong to the same group, in principle doing the same thing at the biochemical level, but they are not as incompatible as the first drug was with the first one. And this is the example in case is omeprazole that is being substituted by other drugs that has less incompatibilities. Now the question, the interesting question on this is how fast the system is adapting to the new drugs, how much this cost in terms of economical cost, and also in terms of the education of the doctors and the patients to change the medication. That has to do a lot with what people believes in previous drugs and all that. So there are alternatives. So even if we see a number of counter-indicated drug combinations, there are alternatives that has to be implemented as soon as there are new drugs replacing some of these double medications. This is how the different drugs of the same chemical group are progressively being replaced by drugs of the same group with less consequences for the co-administration with other drugs. So I finish here. I told you a few things about comorbidities, the importance of comorbidities as a biological and a social medical problem. The more we see how the medical systems are working, and the population is getting older, the more comorbidities we will see. So this becomes a socioeconomical problem. Therefore, it's interesting and important to understand how the diseases are related following the medical records. And if you want to do that, and if we want to understand this problem well, we have to be very careful with doing this, taking into account the covariables. And the two main covariables, the two more important ones, are obviously the gender and the age. And I'll show you some of the data, how the different are the comorbidities once that the covariables are taken into account. To be really able to intervene in the system is not enough to observe the comorbidities at the medical level. Is essential to understand the molecular basis. Part of this molecular basis may be related with the physiology, and one way of assessing physiology is the level of the expression of the genes. And we demonstrate, I think that I'm convinced that we demonstrate this properly, that the levels of expression, the expression profiles, can be used to map some of the molecular basis of diseases, at least for many of the diseases or many of the comorbidities that are observed at the medical level. This number of 50% of the non-diseases or the non-correlation between diseases may have a trace at the molecular level that can be interpreted in terms of fundamental biological processes. More intriguing and good for discussion is the other type of correlation between diseases that are related not with the level of expression, but with the presence of variable genes. Why some correlation between diseases are more related with the presence in the population of genes of variable level of expression than with the level of expression itself. I think it's an intriguing biological problem that has to do with the biological interpretation of the variability of these genes that have a sustained variable level of expression. With all that, the other alternative explanation is what is the impact of drugs. I'm not showing that, but I show you that we enter in this area of drugs. And with the first study, pretty consistent between very different populations of how drugs are co-administered depending on gender and age, and why the many examples of pairs of drugs that are given together to people for probably good medical reasons, maybe in some cases not so good medical reasons, and from this will be important to understand what are the molecular consequences of this co-administration, and in practical terms will be important to find alternative, not to have to give drugs that we know are not good in combination. So I finished with that. I showed you the picture of the Barcelona Supercomputing Center. I would like to invite you to visit. Indeed, I can invite you to visit as soon as you can do it, because it's a nice place to visit. We are looking for students and postdocs, so you are interested in this area of comorbidities, of course, but we are making other things that are related with genomics and artificial intelligence, or disease artificial intelligence, simulations at the level of cells and at the level of systems, pathways, or at the level of cell-cell interactions, or doing natural language processing. Indeed, it's a big place, so we are doing many different things in different areas of computational biology. So you are most welcome to visit. And if you are interested to not only visit, to stay and work with us, you are also very much welcome. I'm going to, because I see that Beatriz and John are here, so I showed you the first picture, because if there are difficult questions, I may relay some of them to Beatriz and John. Beatriz has done and they have with John and other people, but she's the main author of the papers on the correlation between diseases at the molecular level, at the level of expression profiles, and also this intriguing work of the variable genes. And John has done the work on the CD app, medical records, and also this work on the drug-drug interactions. Thank you.
So thank you very much, Alfonso, for this beautiful introduction to a very, very complex world, I would say. And then I will leave the platform to Gigi for handling the discussion, and the discussion is open.
Thank you very much, Alfonso. There is a first question in chat from Professor Capranico. I don't know if Giovanni wants to add something, but the question was, which tissues have been analyzed in RNA-seq measures?
Yeah, that's something I should have said before. Obviously, we take whatever is in the data set. In some cases, say tumors is easy because there is already a control. In some other cases, the controls are less clear because there's just blood samples, and many of these are taken from. I would say most of the data sets are from blood samples, and you can do a subsection only with the blood samples, and they reproduce the core of the observations.
Thank you. I invite other people to ask questions. The raising hand is in the reaction menu of Zoom, the lower bar or the bar, wherever it is in your system. There is a reaction, and if you open, there is the possibility of raising hand. There is a question from Giulia Babbi from the Biocomputing Group.
Hi, Professor. It was a great talk. Thank you very much. And I have a general question. So in an era in which we are going in the direction of, many are going in the direction of precision medicine, should we before revise the definition, the fundamental definition of what is a disease, or better, how far we are from a new classification of diseases based on the pattern of similar pathway and gene expression, somehow like what molecular-based approach have done for bacteria and microbiome classification?
No, that's exactly the point. I cannot agree more with you, Julia. When we take the expression profiles and compare them between different individuals and cluster all them together, what essentially we are doing is reclassifying all these patients in different groups or subgroups, what Beatriz called the meta patients, that in principle have nothing to do with the diseases. All the information that has been used to classify them is the expression profile. Then later you came with a label and say, they were classifying this disease, but they are not all the cases that belong to this disease. It happened to be that all of them are classifying this in disease, at least at a given level, but then they have different levels of relation with other diseases. So there's a subgroup of patients that classify with molecular information, as you suggested, And this classification makes them unique in terms of the interaction with other diseases. And the disease label is something that came later, came at the level of, at the time of labeling them, not at the time of defining the groups. So it's exactly toward this idea.
Thank you. I invite all the people to raise the hand and ask question. The next question is from Professor Casadio.
Yeah, I'm rather curious now, since you entered into the details of understanding why women and men are so different in reactions sometimes. And then, I mean, perhaps you cannot answer because it's a very detailed questions. But which are the most variable genes among men and women?
I don't know. That's a good question. Beatriz, do you know?
Now, I'm very curious because, you know,
we have the expert in the room.
The other
Beatriz.
question, if I can rephrase it, is: do you have any hint about the difference among women and men? So the gender difference in relation to the different reactions to diseases and the different reaction to drugs, possibly.
Yeah. I leave Beatriz to I don't know, the genes that are more variable. I don't know. Do you know, Beatriz?
Sadly, we do not have this information in our data set. So we are using publicly available data sets. So we don't have the sex of most of the samples. So we couldn't explore this. But I'm sure that with other data sets, maybe we can take a look and see what happens.
So we have not done the gender for the gene expression.
It's going to be really important, you know.
But for the reaction to the drugs, at least for the co-administration of the drugs, then there are plenty of examples, because obviously the biological processes are very obviously different. John, you want to give one example? You are the master of the examples of the drug interactions.
The microphone is
John, the
muted.
mic.
Sorry, and I lost part of the question, Alfonso. Can you repeat it, please?
Examples of influence of drugs co-administration in males and females. What is your favorite example? The different consequences.
Whoa, so I mean, more than the consequences itself is clearly the fact that the fact that different males and females have different disorders. This will derive to different drug-drug interactions. So for example, there are things like the interactions of drugs to treat osteoporosis and the drugs that are treated to avoid or reduce the pain associated to osteoporosis. These two drugs are known to interact, and this is something that you will observe a lot in males, but not also in females, but not in males, as the incidence of osteoporosis is really small in males. And this is indeed clearly associated with aging, as people that are, females that are older than 50, the incidence of osteoporosis is quite high.
So thank you, but may I ask you something else then? So after all, being human implies that you will end up more frequently with osteoporosis. So in spite of the fact that males live shorter than women on average, then you have the advantage of not ending up with osteoporosis.
Well, indeed, in this case, it's not just that. So I mean,
I'm kidding.
This is corrected for this. It's more on the hormonal changes in females.
Okay.
It was just joking. Yep.
Thank you very much.
Thank you. There is a question in chat from Francesca Catalonia, that is a student of the master. How has it been shown within the expression profiles that there was a particular decrease in certain diseases as a protective factor? Could it not be incidental? I don't know if Francesca can add something to the question, opening the mic.
Yeah, hi. Yeah, You might have mentioned it, but I was just curious how we can prove that there is a quote unquote protective factor, because could it not, I mean, we haven't really scoured every single patient, and how can we prove that that is the case?
I hope I was careful enough to not to say that we have proved that. All what we see is that there are cases at the medical level. There is a clearly anti-correlation between the diseases. My favorite example, because it's striking, is schizophrenia and lung cancer. Patients of schizophrenia have clearly less chances of having lung cancer, even if they are heavy smokers in average. This has been done in many different population studies with the controls, the age, the something. So I think it's a pretty, I would say, it's the kind of thing that is well known, the Lancet type of well-known thing. What you see at the molecular level is that there is an anti-correlation in the expression profile of some of the patients of schizophrenia with the corresponding expression profiles in the tumors. I think that genes that are highly expressed in one are downregulated in the other way, and the other way around, an anti-correlation. And this is only true for part of the patients of schizophrenia. It's not true that it doesn't mean that all the patients of schizophrenia will be protected from cancer. Some particular cases of schizophrenia apparently are protected from cancer observation, and apparently there is also some group of patients, and they are not the same because we don't have the same samples, have this anti-correlation. Suggestion that this anti-correlation may be on the basis of this protection, of this medical observation, but we cannot prove that. Indeed, we spent quite some time thinking how to prove that, and it's not a trivial thing. You will need, first of all, you will need to have the samples of this population, of these cases of schizophrenia that do not get cancer, and samples of patients of schizophrenia that do get cancer in enough quantities. They make the expression profiles and find that they are indeed anti-correlated. What is, a nice thought experiment, but difficult to do in practice.
Right.
And then, well, because you have to find the population, find the samples, follow them in quantities, and do the experiment. And we discussed this with not with schizophrenia, with Alzheimer, because there are many groups working in Alzheimer, but it's something that will take a lot of time, because you have to wait for them to develop or not to develop another given type of disease. And we try to do this with Alzheimer, but imagine that you do that and you find that yes, there is a correlation. Nice, you can do some nice biology on that. It still will not prove the point that the anti-correlations means inverse comorbidities, because you just show one case. So really, to demonstrate that the correlations are related with the inverse comorbidities, you will have to do a few hundred random cases, and that's a little bit difficult. So I think that you are touching a very important point, that demonstrating things that are done at this level of genomics, comparing data, and making inference to the phenotypes is a pretty hard task. And this goes to with the significance of GWAS and the other population studies that are difficult to translate to some other molecular levels.
But just to clarify, so there has been shown a downregulation in correlation with, say, schizophrenic patients getting lung cancer. There has been shown molecular.
No. There are two different. Let me see. Yes, the two things has been done. Let me try to explain this clearly. There are two different observations: the medical observation with a cohort in different places of the world, and then the molecular observation with the data from the databases at the level of expression. We don't have data, there are no publications of expression profiles of patients of schizophrenia with and without cancer. Nobody has done this study. It's a complex study to do.
I find it to be a little redundant, because couldn't it just be a genetic component in saying that these patients, we have a pool of schizophrenic patients, and their incidence of, say, for example, lung cancer is not as prominent, but couldn't that be the case with almost every disease? And isn't it a little redundant to study something like that, because there's no way you can ever prove something that way because you have the genetic component involved? It just doesn't seem to.
Well, no. Well, the way I see is that if I can do, can I understand? Can I use this to understand the basis of the two diseases? Can I find a set of genes or a set of pathways that are related with these potential relations? So they are anti-correlated at the expression level, and we think that they are in a group of cases that at the same time are correlated with the observation. Can I understand this and use this to understand the basis of the molecular basis of the two diseases? And can I translate this to a model, animal model or molecular model, in which I can prove that indeed these genes are related with the characteristics that are expected in these diseases? So that will be an operational model to use this information, even if I cannot prove at the population level the correlation. So I think that will be this what molecular understanding of the diseases imply. If you want to think more in the future, you can also think that imagine that you repeat these studies and you convince that the correlation is strong enough to be significant. We have done that, but imagine that is repeated with many other data sets and all that. Then this could be used as indications for the treatment of the diseases. You are a patient of whatever disease, and you know that you are going to have a higher risk or a lower risk of another disease, then it's something that you may be able to prevent with drugs or with behavior, or just by following these people more carefully. So think, the way I see it, at least, and this is not very different of what we do with the rest of the molecular profiles of diseases, only that this is applied to correlations. You think the profiles for the example of breast cancer. All what we know is that there are profiles of people with breast cancer that are correlated. And then you do a GWAS and you are more sure that there is a correlation. You are statistically sure, the sure you can be with the statistics of the correlation. And then you use these markers to do prognosis of the diseases. In this case, we don't have as many observational studies as in the case of subclasses of breast cancer. But for most of the diseases, this is the case. We don't have that many observational studies, and we start observing correlations. We try to confirm the correlations and use these as markers. So I think it's the beauty and the weakness of all these studies.
Exactly.
Thank you. Thank you. There is a question from Elisa Bertolini, students of the master. Elisa, you can open the mic. Bye.
Yes, so thank you. Hope you can see me.
Yes.
If I may, I have 3 questions. So the first question is, do you have evidence on these studies that there is a master regulator, or there are more mechanisms at the same level coordinating to each other, but without a master regulator? And second question related to this, and then I will hold for the third question, is in case there is this master regulators, are there, does this mechanism change in different subgroups? So let's say if we're looking at female through the lifespan, or between women and males. So if we find, let's say, a key pathway, do we have evidence that a key pathway may change?
Yeah. Very good point. You open a research line in here that we will very much want to follow, but we need some people to follow that. So I'm back to if you are interested, you are most welcome. No, it's a fundamental question. Now we have this, I would say, pretty large set of genes, correlations between profiles, mapped to pathways. Now the question is why this happened, no? And what is how the system is organized? Are there master regulators in the sense of regulation? Are the mutations related with the expression? Are these genes overexpressed because they are induced by a mutation in a promoter that is what caused the correlation between these two diseases? is there a mutation in a transcription factor that is controlling part of these genes, and this is what is down this what you call a master of the system. All these are very good questions. I think that answering some of these questions in a concerted way for a number of these pathways will make far more clear what is now, what are the true possible true genetic origin of some of these correlations. And in principle is something that can be done. No? Now you have the genes, you can reconstruct the networks, you can reconstruct the gene expression networks, you can reconstruct other type of networks and try to understand what is the logic of the system. But obviously it has to be done, and we have not done it.
So thank you. If I may, the third question, so it was about, so it's, I think it's an enormous source of information that we have, and it's precious for us to retrieve some data that are online. But these data are based on some assumptions, and then we run statistics and we run indicators of statistic significance based on some assumptions that are underneath the data that are published. So my question is do we have enough data to run statistical significant indicators that are reliable and that accomplish to the assumption? So I better explain. So my question comes from this article that I was reading, and it was regarding a master regulator. So the master regulator, it was behaving on stress conditions, heat stress conditions. So it was on plant and not on human. But basically, if the stress was short term, the master regulator was acting as a positive master regulator. If the stress was long term, it was reversing its action. So it was acting as a negative regulator. So my doubt now is this. So if we take drugs, for instance, and drugs to patients, are we sure that we're testing the same amount of drugs for the same length of time in the same conditions, so we can expect reliably the same results in terms of pathways?
Really, no. No. I mean, humans and there are many. I take this. Let me take this in a different way. The amazing thing is that we can still find trends and of things, and we can still build consistent data and build hypotheses on that, given the type of data that we have. Because you think on that, we have expression data that was done in different individuals, in different tissues, with probably different techniques, and with in general, with very little information about how the experiments were done. And still, we, not us, but in general, all the use of this expression data has provided a lot of clues about systems, diseases, pathways, functions, etc., even in the middle of all this noise for experiments. And this is applicable to the rest of the biology. If you think how the information is collected in the medical records by different doctors in different conditions with different coding for different people, etc., it's amazing that anything can be extracted from that, and still, things are extracted from that. If you think how clinical trials are conducted with 20,000 people now that we are talking about vaccines, different people, different conditions, and still the methods and the statistics are powerful enough to tell us that this vaccine is going to be safe. So I tell you any field of biology and you will find the same thing. The data we have is very little, very heterogeneous, very noisy. The data we will need to have, if you think as you do in ideal conditions, you need to know that this individual is isogenic to other individuals and is taking the same drug in the same conditions, and you can only change a few variables. That's not what we have. I take it on the positive side. Despite all that, we are doing progress. Biology is undoubtedly doing progress, or computational biology is undoubtedly doing progress, despite the lack of good the quality of the data we want to have.
Thank you very much. And that is it from me. Thanks a lot also for the
Thanks to you for the questions.
Thank you, Elisa. There is another question from Gabriele Vazzana, student of the master.
Yes, good morning. Don't know if you can.
Good morning. Yeah, we see you.
So, Professor, in your lovely presentation, you have briefly showed us the network of the comorbidities, diseases, and how it changes during the aging. And I was wondering if these changing in the path in the networks are related more in the changing of the partners of the disease, so disease change partners with age, or just establish new relation with other diseases, for in the so-called, if I'm not mistaken, you called it multi-morbidity or multi-comorbidity, or if this change of pattern is more related to the insurgence of more frequent diseases with aging, or it's maybe both of them.
I would say that there's a combination of both of them. There are more diseases that appear with age, and that's very clear. And then there are many more connections that appear too. So the network is, I would say that the main thing is increasing in density. It's increasing in the number of connections. But then the number and the type of diseases, they also change a lot. I mean, you go by ICD codes, there are diseases that disappear with age, and then others appear. But the main thing is the increase in connectivity.
Okay, thank you.
Thank you. Francesca, Catalonia has another question. I invite other people to participate. Francesca.
Yes, I just wanted to ask, in your opinion, what are some obstacles within medicine that you think are feasible to overcome within this generation to propel what we need to explore within these expression profiles and finding the clear distinction with age and
yeah, that's a good question. Thank you. For me, this is very clear. The main obstacle right now is the access to the medical information. The way the systems are organized, there are 17 different medical regions in Spain. There are 20-something in Italy. They don't share the information. Each one of them is organized in a different way. Building a project that will do something as we have done for Catalonia. You want to do it worldwide, or at least European-wide. You want to use the medical record from the different regions to see how the comorbidities, the drugs, the symptoms, etc., etc. The main problem you will have is that there is no way to access this information. No way. You have to fight a lot to get the one of Catalonia. We got this one, very nice, but it's only 6.5 million people. There are another 17 regions in Spain that will be good to analyze, and then you go to the other country. So this is a very serious problem. Access to the information, open access to the medical information for research under the security conditions and all that. And they've been a lot of talking. There are big European projects, the European health area, etc., etc. And this is still the main problem. Once that you solve the problem of access, then you will find all the technical problems, different coding, different way for describing things. The codes are not enough because you want to know the symptoms. The symptoms are in free text. You have to pass this to some other type of codification that will allow you to operate with information. And then you enter in the scientific problems, how to analyze this information, how to put it together. And once that you finish with that, then you think, but then we have to add to this the genomic information. Do we have enough genomic information? There is this European initiative the 1+ million genomes that is trying to put together the genomic information that has been generated in the different countries. It's taking a while, and how the system, a federated system to access the genomes that has been gathered in different countries is going to work or something is still work in progress. So these are the obstacles: access the information, being able to understand and use the information from the medical side, but then linking this to enough genomic information. You know, you see this with the virus and the SARS-CoV-2, no, with the coronavirus. The number, no, we need sequencing. We need to know if there are new variants. Everybody is very worried about the new variants. The countries have difficulties to get to a 2% of the cases sequenced. 5% will be the minimal done. The UK has done more than 600,000 cases. Spain has done 10,000. So, you know, the obstacles are, in the case, once that you get the medical information, to get enough genomic information, meaning sequencing genomes plus gene expression information, is a challenge.
Thank you. This was a wonderful presentation.
Thank you.
Thank you. There's another question from Federica Veneziani. I think she's a PhD student.
Yeah, PhD student of Bologna. Yes, thank you so much for your talk. I have a question. How much you're within the same condition, medical condition, how much your gene expression is affected by the different drug that could be prescribed for the same condition, and how much this affect your comorbidity and your comorbidity analysis?
No, that's I said, this is something that we think that one of the reasonable causes of comorbidities, positive and negative, are drug administration. It is it's a good hypothesis. It's very difficult to prove because in the gene expression data, in many cases, the information that is associated, the meta information, is not very good. So you don't even know what are the drugs that people have taken. So it's difficult to deconvolute this in terms of. So it's not that, you know, for the given gene expression data is not that you have the associated medical history of this person with all what these people have been doing. It's not that at all. Normally, you only have a few annotations of the origin of the data. You know, the first cancer genome project, the International Cancer Genome Consortium, that put together this huge program to analyze cancer genomes and was very successful. When you go back and say from these samples, these cancer samples, how much you know about the conditions, the drugs that people was this a tumor, a primary tumor, taken before or taking some drugs before of any conditions, the information is not there. So the full project was done without knowing, knowing only the cancer type, breast cancer. So we are lacking annotation. Now the second phase of the project is done, putting more care in the annotations in a way that we know the origin of the sample, the type of cancer, But again, back to the previous questions, we are missing a lot of information, including the one that you mentioned. What is the drug that is associated to each one of the profiles, if any, and therefore being able to study the influence of the drugs in the profiles.
Thank you. More questions? We have some more time. If someone has some curiosities, some concerns, some discussion, we are pleased to ask. If not, I thank you, Alfonso, again very much for this beautiful presentation and the beautiful discussion that followed. Thank you very much. It was a pleasure to have you even online, but I hope we will see soon, very soon in Bologna or somewhere else.
Yeah, we will try to bring you back to Bologna.
Yes.
Okay. Well, you are most welcome in Barcelona too, and very, very good to see you all.
Congratulations for your work, the development of the work and everything.
Bye.
Bye-bye. And
Bye.
obviously, if you have any question, just ask me by email.
Yes, I invite everybody to join for the next lecture on Thursday at 3 in the afternoon. I remind you that the lectures are meant to start on time, so I will open the room a quarter of an hour before. The next lecture will be given by Silvio Tosato of the University of Padua and will be entitled Bringing Structure to Protein Disorder, Understanding a Biological Phenomenon. So see you next Thursday, and thank you again.
Thank you.
Thank you.
Bye.
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 Thank you, bye.